Hello everyone, welcome back to another video tutorial. Today we will see some important terms of genetics. The term genetic was coined by Bateson in 1905. In terminology of genetics, at first we see the term gene was coined by Johansen in 1909. At first we will see about gene. So, gene is a functional unit of inheritance. It means, as per Mendelian concept, the pair factor that control a particular character is called gene. For example, the trait of plant is tall. The tallness of plant codes capital T and capital T, whereas trait of plant dwarf, for this it codes small t, small t. So, as per molecular concept, gene is defined as segment of DNA that codes for a particular polypeptide. Now the allele. Gene consists of allele, which is also known as allelomorphs, or also can say that alleles are alternate form of a gene which occupy identical loci, as you can see here, identical loci on homologous chromosome. And allele also control the contrasting character of same trait, and usually allele exists in two, that is, for example, the dominant and the recessive one. Here capital T is dominant, whereas small t and small t is recessive. We always use capital letter to show dominant and small letter to show recessive one. Now let's see what is dominant and recessive trait. The dominant is the allele of a gene that suppressed the expression of recessive alleles and will always express in F1 generation after a cross is called dominant and are classically symbolized with capital letters. For example, here for the purple color flower, we always used capital P or also capital P and small p, but it is in homozygous condition, whereas it is in heterozygous condition. Then the next one is recessive. Recessive is an allele that is masked by a dominant allele, does not appears in the heterozygous condition and only appear in homozygous condition. We can also say that the character which are not expressed in F1 generation after a cross is called recessive, and recessive is always written in small letter. So for example, here for white flower we always use small p and small p, which is in homozygous condition, not in heterozygous condition. Now the next is homozygous condition. So it means when both the allele of a particular trait are of same type, as you can see here. Or we can also say that a condition in which both the member of an allele pair in a homologous chromosomes are identical, means either dominant or either recessive allele. For example, here it is homozygous condition. It codes for either tall, that is capital T and capital T, or dwarf, that is small t and small t. Now heterozygous condition. When both the allele of a particular trait are different, as you can see here, both are different. Or we can also say that a condition in which both the member of an allelic pair in the homologous condition are not identical, that is one dominant and one recessive allele. As you can see here, it is in heterozygous condition. It is one dominant and one recessive. So now we will proceed to the cross. Cross is a breeding of two plants. For example, if we do cross between the plants having the trait tall with dwarf one, it will be in homozygous or heterozygous condition. So cross is made between one, two or more than two traits. First one is monohybrid cross. So when a cross made between one pair of contrasting character, Such type of cross is known as monohybrid cross. Or it also states that an organism which is a heterozygous with respect to only one pair of allele at a locus under study. For example, as you can see here that pea pod color is yellow crossed with green pod colored pea. So it shows the monohybrid cross. Now second one is dihybrid cross. So when the cross is made between two pairs of contrasting trait, such type of cross is known as dihybrid cross. Or we can also say that an organism which is heterozygous with respect to two pairs of alleles at two loci under study. For example, here yellow colored and round seed is crossed with green colored and wrinkled seed. In the same way, trihybrid cross done between three contrasting character. As you can see here it is round, yellow and full seed is crossed with wrinkle, green and constricted seed. So here it takes three trait for cross, which is called trihybrid cross. Now move to the self cross. So when F1 generation is crossed with F1 generation, such type of cross is known as self cross. As you can see in this picture, after getting this F1 generation, it is self crossed here. So it shows self cross. Now the next one is test cross. When the F1 generation is crossed with recessive parent, such type of cross is known as test cross, and it is used to determine whether the individual exhibiting dominant character are homozygous or heterozygous. As you can see here that after crossing parents, we get the F1 generation, and then this F1 generation is crossed with recessive parent. Now the another one is back cross. It means when the F1 generation is crossed with either of parent, called back cross. One more thing, if F1 is crossed with dominant parent, all progenies, that is F2, will be dominant, whereas if F1 is crossed with recessive parent, then individual with both phenotype will appear in equal proportion, and the ratio of progenies produced during back cross is called back cross ratio. And here you can see that after crossing the parent, we get the F1 generation, and then we cross the F1 generation with either of the parent. Here we take dominant one, so it shows the back cross. Now next one is reciprocal cross. It involves two crosses concerning the same characteristics but with reversed sexes. As you can see here it is female crossed with male, and if the trait are autosomal, the reciprocal cross always yields same result, and if the trait are on sex chromosomes, the reciprocal cross give different result. So these are about the cross. Now pure line. A pure line is a progeny of single self-fertilized homozygous plant. What does it mean? It means when we have a particular genotype being self for more than 5 consecutive generations for the purpose of getting the plant which shows only one type of genotype. That means if there is homozygous condition, all should be homozygous dominant. As you can see here, parent generation is crossed, then we get F1 generation, and we will repeat the same process for at least 5 times to get pure line. Now proceeding to another topic, that is parental generation. So the generation from where any cross begin. It means we know that any cross started with parents. So this stage is called parental generation. And now the generation obtained after crossing the parents called filial. For example, F1 generation, F2 generation. So what are these F1 and F2 generation? So at first, F1 generation is the generation obtained after crossing the two parents. It shows F1 generation. That is, it shows only dominant characters. Whereas in F2 generation, the generation obtained after crossing F1 generation, phenotypic and genotypic ratio are same in F2 generation. Now, see the phenotypic and genotypic character. So, a pure line is the progeny of the single self-fertilized homozygous plant. As you can see here, it shows the phenotypic characteristic. Or we can also say the morphological character of an organism known as phenotypic character. Whereas genotypic character is the genetic makeup of an organism. Or we can also say that the genetic contribution of a character known as genotypic character. As you can see here, this one shows the phenotypic character, whereas this one shows the genotypic characters. Now the last one is punnet square. So it is a grid that enables one to predict the outcome of a simple genetic process, or it gives the F1 generation and represent the probability of the genotype and phenotype of the next generation. So this grid shows punnet square. So from here we determine the relative proportion of genotype and phenotypes of the offspring, as you can see here. So all these are the basic terminologies related to genetics. I hope you all understand these terms very well. See you soon in the next video tutorial.